forensic chemistry today, we must first look at its origins. One of the earliest breakthroughs came from James Marsh. It called the Marsh test. It was created in 1836 that became the first reliable method for detecting arsenic in the human body. At a time when arsenic poisoning was common and difficult to prove, this test provided a clear scientific evidence by providing a visible metallic deposit when arsenic was present. Its introduction significantly improved the ability of courts to convict poisoners and marked a major breakthrough in forensic toxicology. Next, in the early 20th century, Edmund Lockhart established a foundational concept in forensic science known as Lockhardt's exchange principle, which states that every contact leaves a trace. He also founded the first police crime laboratory in Lyon, France, promoting the use of scientific methods in criminal investigations. His work emphasized the importance of trace evidence such as hair, fibers, and fingerprints, helping shift forensic science away from reliance on confessions toward objective physical proof.
History of forensic services in the Philippines. The development of forensic services in the Philippines evolved from basic medical legal practices during the Spanish period to modern scientific crime laboratories under national agencies. During the Spanish era, forensic work was limited to legal medicine, where physicians conducted autopsies and determined causes of death for court purposes. There were no formal forensic laboratories, and investigation relied mainly on medical observations. In the American period, modern forensic science was introduced. The establishment of the Bureau of Government Laboratories marked the beginning of laboratory-based forensic chemistry and toxicology, shifting investigation towards scientific analysis. After independence, forensic services became more institutionalized. The National Bureau of Investigation, or the NBI, was created and developed its own forensic divisions. Later, the police established crime laboratories, including the Philippine Constabulary's forensic units and the Command Criminalistics Laboratory Center, strengthening the training and research. In the modern era, forensic services are mainly handled by the Philippine National Police, particularly its PNP Forensic Group and the NBI. These institutions now use advanced techniques such as DNA analysis, digital forensics, ballistics, and crime scene investigations, or the SOCO.
One of the most important principles in forensic science is the Lockhart exchange principle, introduced by Edmund Lockhart. It states that every contact leaves a trace, meaning that whenever two objects or people come into contact, there is always an exchange of materials. This idea explains how evidence is created during a crime, even if the traces are too small to be seen. For example, when a suspect enters this crime scene, they may leave behind hair, fibers, fingerprints, or skin cells. At the same time, they can pick up dust, soil, or other particles from the environment. These tiny pieces of evidence may seem unimportant, but they can provide strong clues about what happened and who was involved. Forensic chemistry plays a key role in detecting and analyzing these traces. Using scientific methods, experts can identify substances, compare materials, and determine their origin. Because the results are based on science, they are considered objective and unbiased. Overall, the Lockhart exchange principles is important because it helps investigators connect suspects to crime scenes and reconstruct events. It also ensures that evidence used in court is supported by scientific proof, which can either support or challenge testimonies and help achieve justice.
In forensic science, not all scientific evidence is automatically accepted in court. There are specific standards used to determine if the evidence is reliable and admissible. The first is the Freire standard. This standard states that a scientific method must be generally accepted by experts in the field before it can be presented in court. In simple terms, if most scientists agree that the method is reliable, then it is accepted as evidence. The second is Doubter standard. This is more flexible approach where the judge evaluates the reliability and the relevance of scientific evidence. The judge considers factors such as whether the method has been tested, reviewed by expert, and if it has known error rate. These standards are important because they ensure that only credible and scientifically valid evidence is used in court. This helps prevent unreliable and misleading evidence from affecting the outcome of a case. For evidence to be admissible, it must be meet the three main requirements. It should be relevant, reliable, and properly obtained. Scientific evidence is also used to corroborate or refute testimony. This means it can support a witness statement or contradict it. For example, forensic evidence like DNA or fingerprint can confirm if a suspect was present at a crime scene or prove otherwise. In conclusion, the Fryer and Doubter standards play an important role in ensuring fairness and accuracy in justice system by allowing only reliable scientific evidence to be presented in court.
Now, we demonstrate one of the most critical procedures in forensic investigations, the chain of custody. This process ensures that evidence remains intact, properly handled, and legally admissible. First, evidence must be carefully marked or tagged with important details such as the case number, date, time, and the name of the person who collected it. Next, the evidence must be properly packaged. Biological samples should be placed in paper bags to prevent moisture buildup, while materials like arson evidence should be sealed in airtight containers to preserve volatile substances. After packaging, the evidence is sealed using tamper-evident tape, and the collector signs across the seal to ensure its integrity. Finally, all handling of the evidence must be documented in a chain of custody form, recording every person who handled the evidence, along with the date and time of transfer. This ensures transparency and prevent contamination or tampering.
The four pillars of forensics. Biological evidence, or the WHO. Think of this as the genetic signature left behind. It's the most personal type of evidence because it involves human biological material like blood, saliva, or skin cells. The reason this is so powerful is DNA. While a witness might forget a face, DNA doesn't lie. It can uniquely link a specific person to a specific spot. We also use things like blood splatter patterns to tell us the physics of the crime, like where someone was standing when an injury occurred. Second is trace evidence, or the invisible link. This is based on Locard exchange principle, which basically says every contact leaves a trace. When a suspect enters a room, they accidentally pick up fibers from the carpet and leave behind hairs or even tiny fragments of glass from a broken window. These are often microscopic. It's like the ultimate silent witness because the suspect usually doesn't even know they've taken a piece of crime scene with them. Third one is impression evidence, or the physical mark. This is all about patterns left in or on surfaces. Imagine a shoe print in the mud or a tire track in the dirt, but it's not just about the brand of the shoe. It's about the unique wear and tear. If you have a specific nick in the heel of your boot, that's a unique identifier. We also look at tool marks like the specific scratches a crowbar leaves on a door frame. It's like a mechanical fingerprint. Fourth one is chemical and toxicological evidence. The how. Finally, you have the evidence that requires a lab to see. This answers the question of how the crime happened. Was there an accident, like gasoline used to start a fire? Was there a specific poison or drug in a victim's system? This type of evidence is crucial for determining the cause of death or the intent behind an act, turning invisible substances into concrete proof. Why it matters. In the end, forensics isn't just about finding one piece of evidence. It's about how these four types overlap. If you have a footprint impression that contains a specific fiber or trace and a drop of blood, biological, you built a bridge between the suspect and the crime that's almost impossible to burn down.